Oh, it's on the wrong screen. Yeah, you guys were you seeing yourselves or just a blank background? Sorry about that. And thank you, Deacon, for pointing that out. So as I was just talking, those things that I was saying were all on this first slide. You haven't missed too much. So sympathetic fight or flight, parasympathetic rest, digest, inbreed, and feed are common short versions of that. So anyways, now that you can see the slides, very important. What was I saying? We're going to mainly be looking at motor control, motor response in the autonomic nervous system. A lot of the sensational response, what you are sensing internally and unconsciously, is going to trigger a visceral reflex to handle that. And then the sensations that do make it up to the brain are going to be processed in the hypothalamus. So with a lot of our somatic sensory pathways, we saw that they almost all had their third order neuron in the thalamus, leading the thalamus, and then sending that signal to the relative part of the brain. The hypothalamus is the control center here for our autonomic nervous system. And so looking specifically at motor control, with our somatic motor control, we just had a 2-neuron pathway, our upper and lower motor neurons. With our visceral motor control, we have a 3-neuron pathway. So you have your visceral motor neuron coming from that hypothalamus, which I said is going to be our main control center in the brain related to the autonomic nervous system. So they are originating, or they have their nuclei in the hypothalamus, and that is going to send a signal to their synapse with the preganglionic neuron, which is either going to be in the brainstem or in some part of the spinal cord. So that preganglionic neuron is then going to send that signal to a ganglia, where it will synapse with a postganglionic neuron. So those names are somewhat nice. So before the ganglia and then after the ganglia, and then that postganglionic neuron will then innervate whatever its effector is. So smooth muscle and cardiac muscle, and then glands are going to be our 3 major effectors. They are also interacting with adipocytes to trigger triglyceride breakdown, release more energy. But we'll mainly focus on smooth cardiac and our glands, smooth muscle, cardiac muscle, and our glands. So 3 motor neuron pathway. The neuron carrying the signal from the hypothalamus that is going to synapse within our central nervous system. So the preganglionic neuron is coming from the central nervous system, either somewhere in the brainstem or down in the spinal cord. And then that is going to synapse in a ganglia. And again, ganglia are essentially groups of neural cell bodies. Right? So they have the dendrites there to receive their signal. So a lot of the synapses that are happening in the peripheral nervous system are happening in a ganglia. Right? You have your groups of neurons with their bodies there, creating that more bulbous looking structure. And then the axons will leave and go to that effector. Right. Oh, no, that skipped a couple. Okay. And so each of these systems both work very similarly in that way with that 3 motor neuron control. And they can work on their own independently on an effector, or and most often collaboratively, working together and producing opposing effects. So where one is going to increase our heart rate, the other is going to decrease that heart rate. And if one is going to cause our blood vessels to constrict, the other is going to cause our blood vessels to dilate. Okay? Those kinds of things. And even within their own systems, they can have inhibitory and excitatory effects on the same structure, depending on what neurotransmitter they're releasing and what receptor protein is there to receive it. So we'll talk about some of those differences too. But again, generally, the sympathetic nervous system is that response to like feelings of higher energy, or things that we relate with being more energetic. So when you are anxious and feeling stress, okay? And stress is not just relating to like your feeling of depression, or that the world is, or you have a lot on you, but like physical stress, like environmental stress of just being in an uncomfortable scenario, that kind of stress. So when you're in an emergency, your excitement, and then the sympathetic nervous system also controls male orgasm specifically. Whereas the parasympathetic nervous system is going to be most active during rest, assisting with digestion, and then for all arousal. So that's why you got that breed and feed shortened version there, referring to the parasympathetic nervous system, has a lot to do with our feelings of arousal and our ability to digest our food. Okay, and I'm not just going to read those responses to you. There's a lot of reference tables in here to use and go back to if you want to see specifics. I guess something I can note on now that I plan on bringing up later in today's meeting. Most of the questions on this third exam are going to be on the first 3 chapters about the nervous system. So you will see more questions about how neurons work and action potentials, how the spinal cord is structured and the peripheral nervous system, and how the brain is structured and the brainstem. Those things are going to be addressed in a majority of the questions. There will only be a couple questions on chapters 15 and 16. So a couple questions related to pathways, a couple questions related to the autonomic nervous system. And I feel like that's only fair to do it that way since we kind of packed in these last 2 chapters here towards the end. But yeah, so spend more time focusing on the specifics of those first few if it's coming down to the wire, because I just realized there are a lot of specifics in this chapter, especially. So don't want you to feel overwhelmed with a lot of that. Okay. So starting with looking at structural and functional things related to the sympathetic nervous system, we'll then look at the parasympathetic and then talk about some of the comparisons, the similarities and differences. So both are going to have preganglionic and postganglionic fibers, right? We had our visceral motor control neuron that's going to stimulate a preganglionic fiber, whether that's sympathetic or parasympathetic. That preganglionic fiber is going to take that signal to a ganglia and stimulate the postganglionic fiber. So we have both, but their length, structure, where they're coming from differs between these 2 systems. So for the sympathetic division, those preganglionic fibers are short. They aren't going to need to travel very far once they have left the spinal cord. They will reach their ganglia quite close to the spinal cord. A lot of them are on either side, like you see here. Some of them are just in front of the vertebra, and some of them are just sitting right on top of our kidneys in the adrenal glands. And so there's not very far for those preganglionic fibers to go, but the postganglionic fibers are long. And with the parasympathetic, you're going to see the opposite. So we also have specific areas of our spinal cord or brainstem where these preganglionic fibers are coming from. So the sympathetic division or sympathetic nervous system can also be called the thoracolumbar division or nervous system, referring to the region of the spinal cord where these preganglionic fibers are leaving. So they all have their nuclei in the lateral gray horns of the spinal cord somewhere within the region of T1 to L2. So that larger middle portion of the spinal cord. So they'll go to their ganglia, synapse with that postganglionic fiber, and then the postganglionic fiber is going to run towards all of these different effector organs like you see here in some of our glands. And something unique to our sympathetic postganglionic fibers is these pearl-looking structures at the ends of their axons, at their telodendria. They have a bunch of these bulbous-looking round pearls called varicosities. And those varicosities look swollen and round because they are carrying a bunch of vesicles that are just full of neurotransmitters. So waiting to be released. So that kind of looks like this. Right? We've got these strings of pearls that are sitting on these smooth muscle cells that it will affect on. And so they're carrying vesicles with whatever neurotransmitter they're going to release. If it's on a effector tissue like this from the sympathetic nervous system, those vesicles are always going to be releasing norepinephrine. And I'm going to say that again when we talk about the different neurotransmitters involved in these things. But norepinephrine is always the neurotransmitter released at the end of our sympathetic division if it's acting on an effector tissue. The only exception is when it is acting from the adrenal gland or on the adrenal gland. That's going to cause the release of either norepinephrine or epinephrine itself into the blood. And like I said, I'll say that again, but this image here is really just to show you that how large and bulbous those varicosities look and why I'm relating it to those pearls. All right, and there's lots of branches, and it almost looks like it completely wraps around this smooth muscle. So it's going to release a lot of norepinephrine to excite those smooth muscles. And so the presynaptic neurons are so short because all of our different ganglia for our sympathetic nervous system are quite close to the spinal cord, where those presynaptic neurons are going to be coming from. So we have our sympathetic chain ganglia, which are what were best represented here, all sitting on the sides of our spinal cord or sides of our vertebrae here. So those just sit on the side. They innervate the effectors of our body wall, thoracic cavity, head, neck, and limbs. Lots of muscles being innervated from that sympathetic chain ganglia, again on the side. We have collateral ganglia, which are sitting just in front of your vertebral column. So they're just anterior, and again, very close. And those will innervate abdominopelvic tissues and viscera. And then our third set of ganglia are found literally within the adrenal glands and their adrenal medullae. So when these are being stimulated, these postsynaptic ganglia are going to release their neurotransmitters directly into our circulatory system, directly into the blood. So some of the neurotransmitters coming from the sympathetic nervous system end up as hormones instead. It's the same chemical. It hasn't changed anything about itself. It's still norepinephrine or still epinephrine. It's the same chemical that would be used as a neurotransmitter, but instead it's being carried in our blood and thus can last for longer, right? Will stay in that circulatory system for a longer time, whereas in a synapse, things are typically cleaned up quite quickly. It causes its response, and some other enzyme comes and helps clean it up, or it's just reuptaken based on the change in voltage, et cetera. But when these are released into the blood, that's a much longer lasting effect, right? As it's traveling and needing to find a receptor protein that's going to take it, et cetera. So some of these are going to end up actually acting as hormones traveling to the blood. And the parasympathetic system does not do this. So only the sympathetic system out of these two options is going to produce a hormone response, epinephrine or norepinephrine into the blood. Okay. And so now we can talk about those neurotransmitters more specifically. So as I was referring to a little bit earlier, the postganglionic neuron that is acting on an effector tissue, so acting on a smooth muscle, acting on the cardiac muscles, acting on our viscera, it is always going to be releasing norepinephrine at the end. All of our preganglionic neurons are releasing acetylcholine in the autonomic nervous system. So for sympathetic and parasympathetic, all the preganglionic neurons are releasing acetylcholine. That acetylcholine is going to interact with receptors that we call cholinergic. So acetylcholine activates cholinergic receptors. And we have two different types of cholinergic receptors to consider. I'm going to have another slide talking more about those. And so in the case of our which cholinergic receptors are sitting on the dendrites of our postganglionic neuron, those are called nicotinic. And like I said, we'll talk about that more in depth in a second. And this is always going to have an excitatory effect on that neuron. And that only lasts for about 20 milliseconds. Like I said, neurotransmitters themselves at a synapse are typically cleaned up quite quickly. So the effect of that signal is going to be carried out, and then cleaned up really, really quick. That postganglionic neuron releasing norepinephrine on an effector tissue or epinephrine into the blood. So really, I guess what's being described here by the 2 to 3 seconds is just referring to norepinephrine on an effector tissue. Norepinephrine is going to react with adrenergic receptors, or both of these, both epinephrine and norepinephrine, are going to react with adrenergic receptors. And that will last for about 2 to 3 seconds. And the reason I'm pointing out the time frame here is then to compare it to how long the hormones last on our blood. Okay? Our blood has a lot less of these other molecules that are required to help clean up those hormones or these neurotransmitters that have become hormones once they bonded with their receptor protein or as they're traveling. And so the response to your sympathetic nervous system coming from your adrenal gland can last for many minutes. So in the textbook, it referred to 30 seconds and then just said several minutes. I remember from psychology talking about this and the trying how long it takes to recover from a stressful situation. Typically, therapists are trying to tell you that you should wait 20 minutes to be confident that whatever reaction you are having has dissipated and that these hormones have been cleaned up and taken off and you are no longer in that fight or flight mode. And so these hormones that get into the blood are going to last a lot longer. And when it is going into the blood, it can also be epinephrine, not just norepinephrine. So it's always any if it's on an effector tissue. If it's coming from the adrenal gland and getting into the blood, it could be either epinephrine or norepinephrine. We also have nitric oxide being released with some of these, and acetylcholine being released onto onto our parasympathetic nervous system neurons. So the sympathetic division can act on the parasympathetic neurons. Basically, if you are in fight or flight, we can also hold off the parasympathetic nervous system from stopping that fight or flight reaction. Okay? So you can inhibit the reaction from the parasympathetic system with acetylcholine while keeping the sympathetic nervous system active. Yeah, and that's our second type of cholinergic receptor, a muscarinic. And like I said, we'll talk about that more in a few slides. So always acetylcholine from a pre-ganglionic neuron, always norepinephrine from a post-ganglionic neuron on an effector, and then can be epi or norepi if it's going into the blood. And so epinephrine and norepinephrine are received by adrenergic receptors. And we have two main types with two subdivisions of each. And so one of these is going to be an excitatory version of an alpha adrenergic receptor. One of these is an inhibitory version of an alpha adrenergic receptor. And then same goes for beta. So our alpha-1, the excitatory adrenergic receptor, so responding to epinephrine or norepinephrine, is much more common. So on our smooth muscles, that's going to basically cause all the things we saw related to muscle contraction happening in that smooth muscle. It's going to ultimately lead to the release of calcium from their sarcoplasmic reticulum, allowing for the binding of the myosin head to the globular actin and then to pull that. And so this is going to cause the constriction of our blood vessels and closure of the sphincters in our digestive tract. Okay? It's allowing for muscle contraction, causing them to contract, so it's going to squeeze those things. Alpha-2 do basically the opposite response. They are inhibitory, so they're going to block those muscles from constricting and even allow them to relax and stretch further. These are much more common where the parasympathetic system is also acting. So again, it inhibits the parasympathetic system from being able to stop what the sympathetic system is trying to do. Okay. Cyclic AMP is just the second messenger. This is a G protein receptor, but we don't need to get into those details. And then we also have beta receptors. So those were alpha, okay, mostly on smooth muscle. And our sphincters and blood vessels. Okay? And then our beta receptors are mostly related to metabolic activity, B1, and play a big role in smooth muscle relaxation, very common in our respiratory system and our bronchioles. So beta-1 is the excitatory. It increases cellular metabolic activity. It increases heart rate, causing your smooth muscles, your cardiac muscles of the heart to contract more frequently. Whereas beta-2 are inhibitory. They will cause our smooth muscles to relax, which is what you're seeing here with the blood vessels. Alpha-1 will cause it to constrict. Beta-2 will cause it to dilate and relax. And they're also in our bronchioles. And so EpiPens as a reaction to like an allergic reaction, trying to ensure that that patient doesn't choke or die, or that they at least have the ability to continue breathing. Right? You're adding epinephrine to the blood. That epinephrine will go and bond with the adrenergic beta-2 receptors in the bronchioles, causing them to relax and not swell up or squeeze as much. And now that patient has a greater chance of breathing and staying alive as they are dealing with this reaction. Okay. So to quickly go back and summarize, run through this again. In the sympathetic division, our preganglionic fibers are really short because they aren't going very far out of the central nervous system, out of the spinal cord. They're all coming or originating from T1 to L2, so this large middle portion of the spinal cord. And then those postganglionic fibers are going to reach out to whatever effector, whether that is a tissue or muscle cell, or if that is adrenal glands. Those postganglionic fibers are unique to the other neural structures that we've been seeing because their telodendria form these really large branching networks with these swollen pearl-looking structures called varicosities full of norepinephrine. The ganglia where you can find the cell bodies of these postganglionic neurons can either be in the sympathetic chain ganglia on the sides of our vertebral column, the collateral ganglia sitting anterior to the vertebral column, or our very specific adrenal medullae, which are within the adrenal glands. So you have two of them, one in each adrenal gland, and each adrenal gland is sitting basically on top of your kidneys. The neurotransmitters being released are consistent across this system. So the preganglionic neurons are always releasing acetylcholine onto that postganglionic neuron. The postganglionic neuron is always releasing norepinephrine onto an effector tissue, but our adrenal glands can release epinephrine or norepinephrine into our blood. But they also have the ability to release acetylcholine to interact with the parasympathetic neurons and some nitric oxide being released. But the main thing to pay attention to is that norepinephrine. So acetylcholine from the preganglionic, norepinephrine from the postganglionic, and then we can also release epinephrine into our blood from the adrenal. So since we are mainly ending with epinephrine or norepinephrine, we're mostly focused on those adrenergic receptors on our effector tissue. So adrenergic referring to these adrenal hormones, epinephrine and norepinephrine. So we have alpha-1 and alpha-2, alpha-1 being excitatory, beta-1 being excitatory, alpha-2 being inhibitory, beta-2 being inhibitory, which play a role in a lot of these functions. So beta-1, you can think of the heart, that's why this little image is here, and its relation to increasing our heart rate when we were in that fight or flight response. Beta-2, you can think of the lungs as, and our relation to the idea we were talking about, like an asthma attack, and needing to relax those bronchioles and provide some more airways or more space for passage of air within your lungs. And then for alpha-1, think about constriction of our blood vessels. Beta-2 also helps dilate those blood vessels, having that opposite response. And that is the sympathetic nervous system. Is there anything you guys would like me to elaborate on or touch back on before I move on to our parasympathetic? Okay. So in the parasympathetic or our craniosacral division, so just like how the sympathetic division, it can be referred to as the thoracolumbar because of where those preganglionic neurons are coming from. Our parasympathetic division is our craniosacral because of where those preganglionic neurons are coming from. So some of them are in our cranium, in our brainstem from the pons and the medulla. And then we have a few down towards the inferior portion of our spinal cord coming from some of our sacral region in the spinal, or some of our sacral, why am I struggling to think of a singular version of what I'm trying to say? But that short sacral region of the spinal cord, S2 through L4. So craniosacral. And they are traveling much farther before they are able to synapse with a postsynaptic neuron or a postganglionic neuron. Okay? And a lot of those postganglionics are extremely short. So even just looking at this image, we can see these are, somewhat moderately long for things related to the head. But if you look at how they are affecting each of these effector tissues of our organs, that black is a postganglionic fiber, extremely short. The preganglionic fiber is basically getting all the way there by itself anyways. So extremely short. And they always synapse in collateral ganglia. So we don't have multiple different ganglia to track here. And so whereas with the sympathetic division, we were saying acetylcholine from our presynaptic, and then we have all these different options for our postsynaptic or postganglionic, sorry. With the parasympathetic division, we only need to worry about acetylcholine. Acetylcholine is being released from the preganglionic to the postganglionic, and then that postganglionic is going to release acetylcholine onto its effector tissue. And so here we can talk about our difference between our two cholinergic receptors. So we can have a nicotinic cholinergic receptor or a muscarinic cholinergic receptor. So these are both reacting to acetylcholine, just in slightly different ways. So our nicotinic cholinergic receptors are basically channels for chemicals to go through. So acetylcholine binds with that nicotinic receptor. That causes the opening of a channel, such as the sodium channel here, which should look very familiar from our neuromuscular junctions. And then that sodium floods in and can cause an action potential, potentially. That's what we're looking at here. Muscarinic cholinergic receptors are G protein coupled receptors. So they are going to activate a secondary messenger, these cyclic AMP molecules, if it's inhibitory, or some other enzymes and proteins, if it's excitatory. And so that's how they have their differing responses, just based on what receptor protein is sitting at the end of that chain. But it's always releasing acetylcholine. So I guess kind of just working backwards to now expand on this. Right? So some of our postganglionic neurons from the sympathetic division can act on the parasympathetic system by releasing acetylcholine onto those muscarinic receptors, right? Activating that secondary messenger to inhibit the function of this parasympathetic division. Right. So this is one of those reference tables. And I think from here on, yes, a lot of them. So just helpful with organizing this information and looking at some of the differences and similarities. Right. And then I really like this kind of summary tables they have split throughout here. So a quick summary comparison. It has different colors of these arrows for the various neurotransmitters that we're talking about. So acetylcholine is always being released from a preganglionic fiber, whether that's sympathetic or parasympathetic. You see the black arrows. So we have acetylcholine acting on our postganglionic neurons. In the sympathetic division, that's always going to lead to norepinephrine being released on that effector target tissue. But if it's acting on the adrenal gland, and this is going to release these neurotransmitters directly into the blood, we also have epinephrine as an option to be released here. Okay? Becoming hormones, taking longer to clean up, causing a longer lasting effect. So you feel the effect of your response to stress for a much longer time than you are aware of or feeling the effect of like a parasympathetic reaction. So in our parasympathetic reaction, it is much more simplified because we're just looking at acetylcholine. So the preganglionic neuron is traveling much farther, almost all the way to the effector tissue, where it will synapse in the ganglia with its postganglionic neuron, and that postganglionic neuron will release acetylcholine onto its target. Another reference table with the structural comparison. I organize these based on like where they show up in the textbook, and that's why we have a figure that I can talk about with a table in between it. But if you download this and want to move all the tables to the end, I would understand that too. Okay. So the next topic brought up is the interaction and these plexi or plexuses. Okay. So dual innervation is the term used to describe when an organ or effector is being innervated by both divisions of our autonomic nervous system. So we have sympathetic and parasympathetic postganglionic neurons affecting this tissue that is referred to as dual innervation, which is an extremely self-explanatory name. So I love stuff like that. Dual referring to two, and innervating. So it's being innervated by both. And we also have multiple areas, or we describe the areas where these fibers are kind of converging and synapsing as their autonomic plexuses. That term, my brain doesn't like that term. I want to say plexi. And I meant to check if plexi is an acceptable plural for that too, plexuses. That's just where these fibers of our autonomic nervous system intersect. So we have a few main plexuses. Your cardiac is that intersection at the heart. The pulmonary is the intersection at the lungs. You can see here. The esophageal is more of just a passageway for these nerves that are going to go affect all these other organs. Right? Our vagus nerve playing a large role in our hearts, lungs, and digestive organs, and the splanchnic nerve. We have our celiac and inferomesenteric plexuses. There's the celiac, there's the inferomesenteric in this area, which is where these nerve fibers that are going to innervate the abdominal cavity are intersecting. And then lastly, our hypogastric, so below the gastric system, below the stomach. And those fibers are going to innervate the digestive system, the urinary system, and the reproductive system within the pelvic cavity. More references. So this is cool, like for your information, not necessarily need to know for the exam, but getting a better breakdown of each of these systems, the organs within them, and how they're being affected by our two different effects, where they're being dually innervated, right? So how the sympathetic system is affecting the blood vessels within your skin, skeletal muscles, heart, and lungs, right? Getting to see that breakdown. Okay. And so now bringing in some of that sensory interaction. Okay. So we don't necessarily have our own separate like visceral sensory pathways. A lot of the sensations that are traveling through these somatic sensory pathways are just going to split off and be taken to the hypothalamus or be immediately reacted into a visceral reflex rather than making it all the way to the thalamus, into the cortex, and causing perception where you are now aware of the sensation. And so for our visceral reflexes, we have something new compared to when we were looking at our other reflexes, where we have this shortcut. They don't always need to go all the way to the spinal cord. So that sensory information is not always making it to an interneuron within the spinal cord that is going to cause that motor response. Instead, that could take that information directly to an interneuron that is sitting within the ganglia. So this is called a short reflex, as it is a shortcut, skipping our spinal cord, causing or allowing for a much faster response, keeping your internal organs functioning or reacting to whatever's going on, right? But we can also have that long reflex, which is a lot like the reflexes we were looking at previously with our skeletal muscles, where the stimulus is causing a response that's going to be carried all the way to the spinal cord. That synapses with an interneuron, and then that interneuron stimulates some motor response, but it's going to require both the preganglionic and the postganglionic neuron to interact there. So it's not just one motor control neuron coming out. We still need to involve both the preganglionic to allow the signal to leave the spinal cord, and then the postganglionic that is going to be innervated at the ganglia. And then again, another cool FYI of talking about all those different reflexes, how they're stimulated, how each of these systems responds. Okay. So a quick summary table that kind of addresses the entire chapter, at least with comparing it to our somatic function. So sensory receptors are going to be involved in the sensation for both visceral and somatic sensation. Your ability to either be aware of it, meaning it's somatic, it made it all the way to the cortex through the thalamus, or something that you're unaware of and is going to be regulated by your autonomic system. So those sensations might innervate into a short reflex, causing a very quick reaction on that visceral effector. They might make it all the way to the spinal cord into the central nervous system, where they will lead to a long reflex of the autonomic nervous system, or a somatic reflex for your somatic nervous system, moving your skeletal muscles. If it makes it all the way to the brainstem, 99% of that information either is going directly into these reflexes or is going to branch into the hypothalamus. So we could say 99% of what is making it to your brainstem is going to the hypothalamus, and that is going to sometimes communicate with the limbic system, sometimes involve information from the thalamus, but that is the main control center. This is our headquarters for the autonomic nervous system, which is going to send that motor response back out and down the spinal cord. If we're talking about our somatic system and what we are aware of, that has to go through the thalamus, and the thalamus will then send it to the relative area of the cerebral cortex. So primarily in the primary somatosensory cortex, which is in the parietal lobes, right, where they meet the frontal lobe. It's just another visualization of those motor pathways, and then another reference table. So let me pull up the study guide and see if there's any more guidance I can give here, or if this points me towards something else that I should spend more time on. Started the scoring for that Patrick Catrala. You started the first. If you guys have any questions in this moment, you are more than welcome to ask those while I'm looking at this.
It's not a question, but how many like questions are we expecting? I see the study guide, like they have like 10 on the top, and I'm not sure, like 3 or 4 on the bottom. So are we expecting all that much from this chapter in the previous chapter?
Like I said, just a couple of questions from chapter 16 and chapter 15. A majority of the questions on the exam will be 12, 13, and 14. So structure and function of neurons, structure and function of the spinal cord and your peripheral nervous system, and then structure and function of the brain. That is going to be a majority of the lecture exam content. Okay.
Thanks.
Of course. Yeah. Okay. Looking at the study guide didn't provide any further clarification for me, which makes sense because I was referencing a lot when I was building this PowerPoint. So I tried to focus and make sure I included the stuff that is mentioned there, and try to leave out a lot of the noise and extra stuff. I mean, all of this hopefully should be interesting to you, even the stuff that we're not talking about in class. And if you're going down this path, taking this course, these things just get harder and more specific from here, right? So if you're not feeling excited about the extra info in these chapters, or not feeling drawn at all to learning more about it, maybe broaden the horizons as far as what species you are focusing on, right? This is all, if you're going to focus on humans, one species, it's going to get really specific. You're going to get into the nitty-gritty, and that should all excite you. But yeah, I left out a lot of the extra stuff. It goes into memory, it goes into sleep cycles. And I'm sure there's more than that, and tried to focus on what is being addressed in the study guide itself.